Hey all, welcome to ShareTrek. This is Raj here, and today's topic is a look at the COVID virus and a new breakthrough. In order for us to understand that, I would like to set the ground first. First, let us look at some simulations to understand what the virus looks like in a conceptual manner. Then we'll see a model created by a supercomputer that matches atom to atom the COVID virus. That will give us an idea of what the virus actually looks like. We'll get an idea of what the spike protein is and how a virus delivers its payload into a human cell. This will help us appreciate the new breakthrough. With that said, let's get started. Welcome back, friends. Here is a model, conceptual model of the COVID virus. It's been conceptualized as a spherical body with a lot of spikes on it. Those spikes are called the spike protein. And inside it is the viral RNA. That the mission of this particular virus is to deliver that viral RNA into the human cell. This here is the human cell. And on the human cell, we have receptors called ACE2, which are this light purple protrusions that you see on the surface of the human cell. And what happens is that when the coronavirus enters the human system, its spike proteins gravitate towards the ACE2 and bind with the ACE2. And once the binding happens, the virus becomes stationary. And at that point of time, these bluish green spikes called TMPRSS2 extend over and they cut through the spike protein, and then they enable the human cell membrane to fuse with the cell membrane of the virus, and that creates an opening through which the virus delivers its viral RNA. So this is the general method by which COVID virus infects humans. With that said, here is a model of what the COVID virus actually looks like, and this has been created by a supercomputer matching atom to atom from the actual COVID virus to develop this diagram. And you can see this is the receptor binding domain, which is at the very top out here. With that said, we have a kind of idea of how this virus infects the human body, what it generally looks like. It's time to dive into the current status of COVID. What is the current status of COVID? How are the vaccines managing to deal with it? And what is this new innovation that I'm talking about, or the new breakthrough that I'm talking about? So let's start by understanding that the therapeutic antibodies that were effective earlier in the pandemic have lost their efficiency as the SARS-CoV-2 has changed and mutated, and more recent variants, particularly the Omicron, have learned how to circumvent the antibodies our system produced in response to vaccinations. This is where research with humanized mice come into play. Humanized mice have turned out to be a promising translational model for studying human immunity. Their utility has been enhanced by the development of new stocks of immunodeficient hosts. Humanized mice can support studies in many areas of immunology, including autoimmunity, transplantation, infectious diseases, and cancer. These models are particularly valuable in experimentation, where there is no appropriate small animal model of the human disease, as in the case of certain viral infections. These stocks of mice lack adaptive immune function, and they display multiple defects in innate immunity and support heightened levels of human hematolymphoid engraftment. Humanized mouse models of immunity refer to normal immunocompetent mice expressing human genes via transgenesis, or putting human genes into the mice, or immunodeficient mice engrafted with human hematopoietic and lymphoid cells or tissues. The model is best suited for studies of immune function from patients with immunological disorders, analysis of antigen recall responses, investigations of allograft rejection, and other short-term experiments which last approximately not more than 4 weeks in duration. And that said, immunodeficient mice should be housed in a specific pathogen-free environment because they are susceptible to infections. They should be in really sterile environments and handled with sterile techniques. The researchers initially introduced 2 human gene segments into mice, causing their B cells to create a wide repertoire of humanized antibodies in short period of time. They subsequently exposed the mice to the original Wuhan HU1 strain of the virus's SARS-CoV-2 spike protein, which is the main protein targeted by our antibodies and by all the current vaccines. The modified mice developed 9 lineages or families of humanized antibodies that bonded to the spike in response. A Duke University team led by Dr. Barton Haynes then assessed the efficacy of these antibodies. Antibodies from 3 of the 9 lineages were effective in neutralizing the original Wuhan HU1 virus. The SP1 cytosine antibody and other members of its lineage in particular demonstrated extremely wide activity neutralizing alpha, beta, gamma, delta, and all prior and current Omicron strains. Many of the existing antibodies work by attaching the receptor binding domain, RBD, of the spike in certain regions, preventing SARS-CoV-2 from binding to our cells. So they prevent the ACE2 receptors and the spike protein of the virus from coming together and binding to each other. The SP177 antibody binds to the receptor as well, but in a completely different manner that does not prevent the virus from binding to ACE2 receptors, but SP177 prevents the virus from fusing its outer membrane with the membrane of the target cell. This thwarts the final necessary step that throws the door open to infection. To me, what this means is that regardless of the COVID virus mutating and changing its spike protein, it will not succeed. This can usher a new class of vaccines that can stop the infections more reliably and potentially put an end to this pandemic of ever mutating COVID virus that we are facing. I'd like to offer a special thanks to Max, one of our subscribers, for bringing this topic to my attention. I found it very interesting, and I hope my illustrations have been able to help make this easy to understand. As you guys know, I'm not a scientist, I'm a generalist, but I found it very easy to follow the concept, and I hope you did too. And if you like this video, please press a like, and it helps YouTube promote our channel, and we'd like that very much. And if you're not yet subscribed, please subscribe and help us grow this channel. Thanks and have a great day. Bye for now.